we present here work in progress to visualize cell motility patterns, out from time-lapse videos. Our method allows to produce a meaningful 2D map containing a spatial organization of small videos of moving cells in a culture, using a feature extraction that characterizes the movement of videos, and a deep convolutional autoencoder. The main motivation for this work is the relationship of cellular motility to cancer development. The genetic alterations present in cancer cells involve changes in the way they move. Indeed, cell movement plays a crucial role in metastatic process, where cells acquire the ability to move and invade surrounding tissues, enter the bloodstream or lymphatic system. Cell movement involves a complex series of events such as cell adhesion, migration, invasion, etc. These processes are regulated by molecular mechanisms and signaling pathways that are strongly connected to cancer processes. So, cell motility analysis can help in several ways. 1. In understanding cancer processes. 2. In cancer diagnosis, since certain movement characteristics can be used as biomarkers. 3. In exploring therapeutical methods by understanding the conditions that affect the cell motility. To map the cell motility patterns, we proposed the following pipeline. First, we partition the input video into small portions, localized in space, 75 by 75 pixels, and time, 16 frames. We then obtain the velocity fields that estimate the displacement of the elements in the picture between consecutive frames. For each frame in the vector field, we compute movement descriptors based on histograms characterizing the direction of movement, HOOF. Then we stack the descriptors for the 16 frames of the small videos, resulting in a 2D image of descriptors that summarizes the movement of the small video. Finally, all the images are projected on a latent space using a deep convey. This latent space represents a manifold that captures the main factors of variation in the small videos, allowing to organize spatially in an insightful manner. The videos used for the experiments were taken from 5 cell cultures with very low levels of SDHB. Cell movements are extremely slow, so time-lapse videos were built taking a snapshot at regular intervals of several minutes each, to produce the input videos. The velocity fields showing the cell displacement can be estimated by comparing consecutive frames. Several techniques can be considered, most of them based in evaluating the degree of correlation between windows slightly displaced from a reference window, and then taking the displacement of that which is most correlated to the reference window. The overall displacement of a region in a picture can be summarized by taking aggregated descriptors of the vector field. The HOOF method considers an easy approach, taking a histogram of the speeds in several directions for the considered region, for example 16 directions. We considered analyzing small videos covering regions of 75 by 75 pixels and 16 frames. The HOOF was applied for the 16 frames to provide time context. Stacking the HOOF descriptors for the 16 frames, we get a 16 by 16 image conveying the evolution of the displacements along the small video. Using a 2D convolutional architecture on the HOOF images, we can efficiently model the spatial coherence and shift invariances in this representation, so we proposed a convey to model the HOOF images of many small 75 by 75 by 16 videos taken from the original ones, trying several configurations. The resulting latent space happened to reveal meaningful groupings of cell motility patterns. We show a detail of the latent space showing some selected videos and how their location in the latent space is meaningfully related to their movement type. This is an encouraging result, showing how cell movement can be efficiently characterized and visualized in meaningful ways on a 2D interface. This suggests further developments in VA tools relating cell movement with other domains such as gene expression data or clinical information that can reveal connections between domains. Here, you can see the computation of the velocity field at different regions of a video. It can be seen that the optical flux algorithm chosen is very stable, and accurately reflects the cell displacement in a wide variety of situations. Finally, we show several snapshots of a demo interface that allows to explore the latent space. In this demo, the user selects several areas in the latent space, and the videos mapped to these positions are displayed. As concluding remarks, we have proposed a method to get a spatial organization of cell motility patterns that can be used in visual analytics applications. The method is still in an early stage, but has given encouraging results for further developments. It has revealed the ability to compare cell motility patterns for a. different cancer types and subtypes, being potentially useful for diagnosis. b. cells with controlled gene alterations, which can be useful in research for new therapies. Being in an early stage, there is a lot of interesting future work, including exploring alternative features for local motility characterization, such as divergence or rotational, used in classical vector field analysis. Exploring the potential for knowledge discovery in cancer processes. And developing visual analytics tools combining clinical and genetic data to discover connections among those domains potentially useful to develop biomarkers based on cell motility. That is all. Thanks for watching.